All right. Good afternoon, everyone. On behalf of the Human Nutrition Research Center on Aging at Tufts University, I welcome our speaker, Dr. Daniel Belsky. Dr. Belsky is assistant professor in the Department of Epidemiology and Butler Columbia Aging Center at Columbia University Mailman School of Public Health. He is a life course aging researcher working in the fields of genetic, genomic epidemiology, and geroscience. His long-term goal is to integrate behavioral and genome sciences in life course research to understand causes of health disparities in aging and to devise novel interventions. His focus for the last several years has been to develop methods to quantify the rate of biological aging in young to midlife humans for the purpose of testing effectiveness of geroprotective interventions that aim to extend healthy years of life. Much of this work is concerned with establishing validity of proposed measures of biological aging and determining the extent to which measured biological aging mediates risk and protective effects of genetic and environmental exposures on health span and lifespan. Dan is PI of 2 large projects involving multi-omics databases for analysis of biological aging, as well as small grants for Russell Sage and Jacobs Foundations investigating how social determinants of health affect biological aging. Over the last 10 years, Dan has worked to integrate molecular measures into ongoing longitudinal studies to test novel hypotheses about disease etiology. He has published over 100 papers on topics in life course, social, genetic, and genomic epidemiology. In 2020, he was listed as one of ISI's highly cited researchers. We are delighted to have him share his work with us today and very much look forward to his talk. As always, there will be a Q&A session at the end, so please post your questions in the Q&A box. Thank you, Dan, and we look forward to hearing from you.
Okay, well, thank you very much, Sai, for the lovely introduction. I'm good and embarrassed now, and hopefully I can live up to to what's been promised. I'm going to share my slides now. And forgive me for my somewhat unusual background. It turns out that if your children's art supplies are behind you next to your white wall, then the Zoom backgrounds end up not looking great. So I just live with this pandemic office, warts and all. So I'm going to talk to you today about testing modification of biological aging in the CALERIE trial. On my way to getting there, I'm going to give you a bit of introduction to how I think about biological aging and how we've worked on measuring it. And then the Calorie stuff is actually going to come at the end. And I'm going to focus mainly on our analysis to test how this trial can inform our thinking about efforts to modify or slow biological aging with the goal of extending healthy lifespan. I'll leave it to Sai and others who are much more expert in the trial itself to really introduce you to or walk through the details. Before I get started, I'll do an acknowledgement in case I run out of time and don't have the chance. Of course, to the support we've received from the National Institute on Aging to do the work, CFAR, Russell Sage, Jacobs Foundation, and most importantly to my collaborators and mentors, Terry Moffat and Arslan Caspi at Duke, my new collaborators at Columbia, Andrea Baccarelli and Zhu Gao, now at Peking University, my co-investigators on the grants that are generating these data, Virginia Krause, Kim Huffman, who leads the Duke site, David Corcoran, who handles the bioinformatics, Bill Krause, of course, who's been just tremendously supportive of all of this work from the beginning and was the person who got me involved with Calorie in the first place. And now Michael Kobor and David Lin at the University of British Columbia, who actually generated the new molecular data that I'll share with you toward the end of this presentation. Here's what I'm going to try and get through in the next 45 or 50 minutes. I'm going to give an overview of a geroscience approach to thinking about our aging population and the health challenges it poses. I'll talk about how I think about aging as a life course process and what that means for how we think about interventions that might seek to modify processes of aging to extend health span. And then I'll get into talking about measurement of biological aging, and finally, how we're studying its modification in the CALERIE trial. So I imagine you've seen these figures before if you've been in the aging business, but if the concept of biological aging is new to you, I'll give a very brief introduction here. We think about aging as a biological process that can be understood as the gradual and progressive decline in system integrity that occurs as we age and which drives disease, disability, and mortality. So aging is many things, among others, a process of human development. That's not what we're focused on when we say biological aging. We're much more interested in this entropic process of decline and decay. And over the course of the past 10 or 20 years, there's been a crystallization in research into the basic biology of aging, in understanding of the molecular basis of this process. And on the left of your slide at the bottom, you see a figure from a paper that referred to the hallmarks of aging. On the right of the slide, there are the pillars of aging, which came out a year later. There are many common elements across these models of the molecular basis of aging, including cellular senescence, metabolic dysregulation, proteostatic alterations, and critically, for what I'll talk about later on, epigenetic changes. But I think what's important to understand is that there are many different components to the accumulation of molecular changes that drive this process of biological aging. And as nicely illustrated in the pillars of aging's image on the right-hand side of your slide, these different molecular components of aging are connected. And so what we observe in aging is really a systemic process that is the outcome of interactions among different elements of our biology. Here's my little cartoon model of what's called the Geroscience Hypothesis. And just very concretely, the Geroscience Hypothesis is that we can intervene on the accumulation of these molecular changes that occur with aging and thereby prevent disease. And that looks, something like this in a little cartoon model where we deliver some kind of intervention. It slows the accumulation of these molecular changes that in turn forestalls or prevents decline in system integrity, preserving function, preventing disease. And there are, in fact, in animal models, now many proofs of concept that this is possible. The older gentleman in the top left of the slide is Elie Metchnikoff, who, among other things, gave us the term gerontology. So he's really just there for the nomenclature. But arrayed around the slide are pictures of therapies for which there is some evidence of effect on the biology of aging through to the prevention of disease, disability, and death. So maybe the most confusing of the images is the Easter Island monolith on the left-hand side of the slide. It's there to represent the drug rapamycin, which is derived from a bacterium discovered in the soil of Rapa Nui. We give rapamycin today to patients who are about to undergo organ transplant to prevent rejection. But in fact, work by Brian Kennedy, Matt Kaberlein, and their colleagues have established that rapamycin affects, in particular, immune regulation in mice in ways that can prolong healthy lifespan. And there are now efforts to translate rapamycin and a family of related compounds called rapalogs so that they are safe for humans and can be delivered in ways that may do things like prevent decline in Alzheimer's patients or possibly ultimately prevent a range of different chronic diseases. At the other end of the spectrum is the drug metformin, which I imagine many of you are familiar with because it's used commonly to treat type 2 diabetes. Metformin has been known for some time to prolong lifespan in laboratory mice. And interestingly, both mice and humans who take metformin get less cancer than those who have metabolic risk managed with other therapies. Metformin is also perhaps the closest to clinical translation. The Food and Drug Administration has approved a randomized controlled trial of metformin to prevent the accumulation of chronic diseases in older adults who do not have type 2 diabetes. And hopefully we'll see the launch of that trial sometime soon. There are a bunch of other things on this slide. I'll orient you to exercise, which is perhaps the closest thing to a molecular fountain of youth that we know of. And then, of course, the empty plate at the bottom of the slide representing caloric restriction, which I'll talk more about as we go on. The other images represent social engagement, which appears to have important effects on healthy lifespan in humans and other animals, as well as the sort of micronutrients represented in the blueberries and the supplements. So there are a lot of different approaches that appear to modify the basic biology of aging, in particular in animal models, in ways that extend lifespan, but we're still at the stage where we don't know the extent to which this translates to humans. And so that's what I'm going to talk about next. I think that for those who are not in the business of aging, and this was certainly the case for me when I finished my PhD, which was really focused on human development, and started doing a postdoc in an aging center where I was starting to think about the other end of the lifespan, is that aging is something that happens at the end of life. Like, we live for a while and then eventually we get sick, it makes us disabled, and ultimately we die. But we've, of course, known for a long time, at least as early as the work of David Barker in the 1970s, that exposures accumulating from the very earliest stages of life powerfully condition our risk for disease, disability, and death at the other end of the lifespan. And so the way we think about aging now, the way I'm trying to think about aging now, is as the biological process linking this accumulation of exposures across the lifespan with the production of the diseases and disabilities we want to prevent in older age. And if that model can be understood as correct, it has some implications for how we think about aging and how we intervene on it. So if exposures accumulate from early life and affect changes to our physiology that precede the onset of disease, because we obviously know that the etiology of chronic diseases extends back far before they're clinically manifest, then preventive intervention actually has to start relatively early, perhaps when people are in their 30s, 40s, and 50s, instead of later on. And in the little cartoon schematic in the bottom right of your slide, what I'm illustrating is this exponential increase in burden of disease that occurs with advancing chronological age. That's the curved upward sloping black line. And just this idea that if we want to rectangularize that curve, which is to say, kind of push the upward slope as far to the right-hand side of the graph as we can, we'll want to begin our interventions before morbidities have begun to accumulate. A consequence is that the timing of the initiation of our interventions will therefore be very far removed from the ages at which those intervention impacts will be most obvious. So we might intervene on people in their 30s, 40s, and 50s to prevent the accumulation of diseases decades later. And the implication there is it would be an awfully long clinical trial if we wanted to try and do this kind of follow-up from midlife preventive intervention. That is precisely the kind of trial, I should be clear, that we run in animal models. In mice, for example, over months, you can observe differences in healthy lifespan between treated and control animals. But obviously the corresponding trial in humans would take longer than we have. So what we're working on is a means to accelerate the testing of these interventions, this alternative geroprotector trial. Sorry, I forgot to introduce the term geroprotector. Geroprotector is a reference to an intervention that modifies the biology of aging in ways that extend healthy lifespan. And so with this accelerated geroprotector design, what we're doing is following our treated and controlled participants for some relatively short interval of time, perhaps a couple of years, and we're examining whether the pace of aging can be slowed in those treated individuals. And this is illustrated on the little cartoon chart on the right-hand side of the slide. Critically, of course, in order to do this kind of a study, we need that y-axis variable. We need a biological age or a measure of the pace of biological aging, so we have a readout on the impact of our intervention. Which brings me to the next part of the talk, where I started almost 6 years ago now with the quantification of biological aging from the physiologies of healthy humans. So again, our question, and this is now some time ago, as we were introduced to the Geroscience Hypothesis and theories of biological aging, and we had in our minds this model of a process of aging as a mediator between the accumulation of exposures from early life and the onset of disease, disability, and frailty at the other end of the lifespan, was it possible to measure this process in young people who were still free of chronic disease? We knew that in older adults, you could track this process from measurements of functional decline, from accumulation of diseases and the kind of frailty indices that Rockwood and Mitnitsky have pioneered, or that Linda Fried developed. But those kinds of measurements weren't going to capture individual differences in young humans. So the theory that guided what we did next, and this is work that happened in collaboration with my mentors, Terry Moffat and Arshan Kaspi, is that aging, if it's characterized by this gradual and progressive decline in system integrity, should be observable from the collective rates of decline in the functioning of multiple organ systems in the body. If we measure any one organ system, we might attribute deficits in function or changes in function to a disease or an injury. But if we could accumulate information from changes occurring across multiple systems in the body, that could give us purchase on this latent, unobserved common cause, this accumulation of molecular change driving an overall decline in the integrity of the system. And if that was happening, we should be able to observe this even in young adults who were still free of chronic disease. So these are post-reproductive maturity, but pre-aging related disease individuals. That's the population that we're focused on here. So just to sort of locate us within my little cartoon diagram, in case that wasn't already painfully obvious, this is where we are at this decline in system integrity phase. And here is the implementation of this theory of measurement. We analyzed data from Moffat and Caspi's Dunedin longitudinal study. This is a longitudinal cohort study of 1,000 babies, all born in the Queen Mary Hospital in Dunedin, New Zealand between 1972 and 1973. So they're all the same age. They've been followed prospectively at the time of this study through their 38th birthday. And the cohort has been maintained with almost no attrition, which is astonishing given that this represented, I believe, the 12th follow-up of the participants. 95% of the surviving cohort members were still participating at that time. And since they were 26 years old, the study had been administering fairly comprehensive health assessments, including blood chemistries, lung function testing, cardiorespiratory fitness testing, blood pressure. There were measurements of complete blood count data. There were telomere length assays. And so we were able to track 18 different indicators that had been measured in parallel when the participants were 26, 32, and 38 years to evaluate whether change was occurring in the many different systems represented by these biomarkers, and if those changes were occurring in a coordinated way. And so what you see in the second panel in the middle of the slide is a graph illustrating just that on average, across this 12-year follow-up period, people's bodies did show the kind of decline in system integrity that aging theory predicted. The biomarkers have been scored here, so they've been standardized by their cross-wave mean and standard deviation, and those that decline with advancing age have been reverse scored. So lung function declines with aging. So FEV1 has been reversed in this graph. The same thing with cardiorespiratory fitness, telomere length, and so on. So what you see is this in this positive manifold is that people's bodies are exhibiting this decline. And in fact, we don't see it here, but when we correlated these changes, it turned out there was evidence of correlation in the rate of change occurring across multiple systems. Now, what we were after were individual differences in this rate of change. And to measure those, we used mixed effects growth models to model for each biomarker how fast individuals were changing relative to the cohort norm. And in this case, in fact, we were using sex-specific reference norms. So how much faster is your A1C increasing relative to the average man or the average woman? How much faster is your cardiorespiratory fitness declining relative to the average man or the average woman? And we then composited those person-specific rates of change to form a single index that we called the pace of aging. And you can see the distribution of the pace of aging in the histogram on the far right of the slide. The pace of aging here is scaled according to the cohort mean. So a value of 1 means that in 12 months, your body changes on average about the same as the average participant in the cohort, or the average man or the average woman. Values greater than 1 indicate a faster rate of change. So some of the individuals were experiencing roughly twice the rate of change of the cohort norm. So they were essentially aging 2 years at a time, whereas others appeared to be aging not at all. So a value of 0 means your physiology looked exactly the same at 38 as it had 12 years earlier when you were 26. Now, because these were relatively young people, it wasn't possible to validate the pace of aging with analysis of mortality. None of the cohort members had died. But the Dunedin study had measured, when the participants were 38 years old, a range of tests of function that would be familiar in a geriatrics clinic. So there were tests of balance, grip strength, motor coordination. There were interviews about physical limitations. There were cognitive tests. And in fact, we were able to compare those cognitive tests to healthy baseline measures that were collected when the participants were still adolescents. So the same test was administered through the age of 13, and then again when they were 38 years old, and we could examine change. And there were also subjective assessments of aging, participants' self-ratings of their own health, and also ratings that Duke undergraduates made of the aged facial appearance of participants. This figure, which summarizes data that we originally published in 2015, shows you that in each case, participants whose bodies were aging faster showed poorer functional integrity across a range of different measures. In a follow-up study, just to sort of flesh out our model of early life adversity or exposure history contributing to a faster pace of aging and on to disease, disability, and death, we were able to use the Dunedin study data to link this pace of aging measure back in time to the exposure histories of participants. And along the top row, you see data for the sort of family history of longevity, the family social class when the participants were children, and their exposure to what the Centers for Disease Control and Prevention referred to as adverse childhood experiences. And so people with longer lived grandparents and who grew up in wealthier families were aging slower in midlife. Those who had more ACEs were aging more rapidly. And we can also see the kind of correlations we would expect with measures of childhood health. So physical health here is a composite of anthropometric assessments, lung function, blood pressure, physician reports about health conditions that children have. There are also the childhood cognitive tests, and then a measurement of self-control composed of a range of interviews with the children and their mothers, and some direct observation and coding of the children when they were very young. And I'm happy to talk about that more during the Q&A. But the bottom line is sort of further proof of concept that not only does a faster pace of aging forecast functional decline, it also reflects the kinds of exposure histories that we know shorten healthy lifespans. So of course, the question was, how does this inform our understanding of this alternative geroprotective trial design? Are we able to put the pace of aging to the clinical test that in fact it's been designed for? And that was around the time that Bill came to me and asked me if I would be willing to participate in the external scientific committee for the CALERIE trial, which of course, I was sort of happy and very flattered to be able to do. And at the time I really thought, well, I don't know very much about randomized clinical trials and or caloric restriction, but I do know something about handling biomarker data. And so what I attempted to do in the service of the Calorie Research Network was take their database essentially for a test drive. And I thought that our measurement approaches were still pretty rudimentary, and it was very unlikely we would produce kind of exciting results. But this was a useful way of putting together the kind of work I've been doing to test what was emerging as a new public resource for the research community. And so I downloaded the database, and I did some work with it. But just to orient you to the trial, before I show you some of their data, this is a report that Bill and his colleagues published a couple of years ago. This is actually published after my first paper on Calorie came out, but I think it's a nice illustration of the sample and its characteristics. So what you can see is there were 220 adults who were randomized 2-to-1 to either 25% calorie restriction or ad libitum regular diet for a period of 2 years. And you can see there was some attrition. but ultimately, most of the folks make it through the trial. In terms of their participants and their characteristics, they're mostly white women, but there are a significant number of men and a significant number of participants who are not white. Calorie excluded participants who were obese, but they did allow participants who were in the overweight range. And so you can see that the average BMI in the trial participants is in fact hovering around that overweight level. Okay. And you can also see in these data that Bill and his colleagues published that the trial had an impact on the physiologies of its participants. And we saw some of these data, in fact, published back in 2015 with Eric Ravison's paper in the Journal of Gerontology, but his figures were not quite as colorful as Bill's, so I used Bill's figures. And it was also not as comprehensive of an assessment of change in the participants. What's noteworthy is that, and I'll show you some data on this a few slides down the road, but as I'm sure many of you in the audience know better than me, caloric restriction is awfully hard. And getting people to stick to a 25% CR diet is incredibly challenging. And so in fact, the average dose achieved in the treatment group is maybe only about half of the dose prescribed. They did better through the first 12 months than they did through the second 12 months. But despite not hitting the target 25% CR perfectly, Calorie did produce substantial and sustained weight loss, and also alterations in physiology broadly consistent with an improvement in cardiometabolic health. So we had seen some of these individual biomarker findings, and that struck me as suggestive that we might also therefore see change in the kind of composite physiological assessment of a person that we were using to understand biological aging. So again, we were analyzing blood labs that were collected for safety monitoring in the Calorie trial at the baseline 12 and 24-month follow-ups. And we were interested in testing this hypothesis that the pace of aging could be slowed in the caloric restriction as compared to the AL groups of trial participants. And these are data published in the Journals of Gerontology a few years back. To measure biological aging, because we only had 24 months of follow-up, we couldn't use the same kind of mixed effects growth modeling we had used in the Dunedin study. There was going to be more heterogeneity between the participants who ranged in age from their late 20s to their early 50s than there was in the rates of change that were occurring over the trial. So instead, we turned to implementation of a cross-sectional measurement of biological age at each of the 3 trial blood draws. And I'm going to show you the data here. We focused on, we borrowed an approach that was first introduced to me by Morgan Levine, who many of you may be familiar with by now, quite famous in her own right. And she had adapted an approach proposed by some Czech mathematicians, Peter Klemera and Stanislav Dubal, for the measurement of biological aging from biomarker data in general. And what Morgan had done initially was to use data from the US National Health and Nutrition Examination Survey to put their model into practice and to test how it was associated with mortality. So what we did in our study was we used NHANES data, and I was concerned at the time about period effects on the biomarkers, assays changing over time, population health changing over time. So we were able to draw NHANES data collected during the same year as the Calorie trial itself was conducted. And using those data, we put together a model of biological age based on indicators of system integrity that were available within the Calorie trial. And you can see that list on the left-hand side of the slide. We pumped them through this equation, which spit out an algorithm to compute biological age from the physiological data. So we essentially took the equation parameters that we estimated from the NHANES, and we applied them to the biomarker data of the Calorie participants. And the result you can see in that scatter plot and the bottom middle of your slide was highly correlated with the chronological age of Calorie participants at pre-intervention baseline. So we have a measurement that is roughly correlated with chronological age. The correlation is about 0.9. There's some variation. And what we're going to do now is ask whether the pace of change in this measure is different in the AL and CR arms of the trial. And what we find is, in fact, they are. So the y-axis in the figure is the change in the biological age. And so if things were working perfectly, what you would see is this straight line for the ad libitum group, the regular diet group, going up from 0 to 1 to 2. In fact, it's a little bit shallower than that. So they age about 3/4 of a year per 12-month follow-up interval. But that rate is dramatically reduced in the caloric restriction group in the trial. So they're aging essentially not at all. Okay. So that's sort of proof of concept from analysis of physiology. It's also kind of old news. So I imagine what you're here for is to understand what we've learned in our new molecular analysis of calorie. And so I'll try to get there as quickly as I can. So we're stepping backward here in our cartoon diagram from decline in system integrity to the accumulation of molecular changes. And just to offer a little bit of motivation for why we should bother, let's recall that the theory here is that aging derives from this accumulation of molecular changes. And what that implies is that molecular measurements are potentially more sensitive to exposure effects. It may be the case that it takes time for changes in the biology of aging to manifest as changes in our physiology. We should be able to detect them first at the cellular level. Molecular measurements may also be more specific. So it may be the case that we're better able to isolate effects on aging from what might be the effects of medicines people are taking, diseases they may develop, and so forth. And then finally, as we think about the long term, molecular measures may ultimately prove to be more scalable than clinical assessments. And this may be counterintuitive at first because you think, well, a lot of these assays are awfully pricey. But prices are coming down, and you can generate massive volumes of data from tiny amounts of sample in fairly uniform ways. So the array technology that we use for DNA methylation, now proteomics, permits the generation of harmonizable data sets on a very large scale to survey sort of potentially a comprehensive view of biological changes in people, much more so than would be involved if we had to take bloods, if we had to do complete blood counts, if we were then doing blood pressure assessment and also cardiorespiratory fitness testing, and we also want to do pulmonary testing and so on and so forth. So, although it may be more expensive today, over the long term, it may ultimately be something that's easier to scale. Now, there are lots of molecular levels of analysis that we could consider. In our own work, and what I'll show you today, we focused on DNA methylation. It was really on the strength of work that was pushed forward, made famous by Steve Horvath and his epigenetic clock. So DNA methylation marks our chemical tags on our DNA sequence. They bond to CG dinucleotides, and they act to regulate the extent to which the DNA sequence is concealed by chromatin and therefore not expressed, or if the chromatin is open and accessible and the DNA sequence can then be transcribed and the genes can get expressed. It turns out that DNA methylation changes occur with aging in a fairly regular manner, such that overall, from maturity at least, DNA methylation is lost as we age at a global scale, but that at individual sites, there are often additions or subtractions of methylation marks. And these occur with such regularity that it's possible to parameterize algorithms that predict the chronological age of an individual from examining only their DNA sequence. And this is what is meant by an epigenetic clock. It's a DNA methylation prediction of the chronological age of the individual from whom the DNA was extracted. It turns out there are lots of these clocks that can be created. They're possible not just in blood samples, but in samples of virtually any DNA, and that you can make them for a wide range of organisms. I believe there's a clock for whales now. So the fact that we can predict chronological age is in and of itself not honestly that interesting. We tend to know how old our study participants are from their birth certificates. But what's interesting about the epigenetic clocks is that the predictions of chronological age aren't perfect. And the theory is that when your epigenetic clock reads out an age that's older than your chronological age, it means you've aged more than we expect for a person as old as you are. And also the opposite is true. If your epigenetic clock is younger than your chronological age, you've aged less. And there were initial and provocative validations of this hypothesis from correlations of the residual of a regression of a person's epigenetic clock age on their chronological age with their future risk of mortality. And there's since then been an evolution in the development of these measurements from pure DNA methylation models of chronological age differences in cross-sectional samples toward what are ultimately DNA methylation models of physiology predicted risk for mortality. And so you may have heard of the pheno-age clock and the Grimm-age clock. These both are based on prediction of mortality, and in between the DNA methylation and mortality is some measurement of physiology. So in the case of pheno-age, there's a clinical biomarker or a physiological model of mortality risk, which is in turn filtered into the DNA methylation data. In the case of Grimm-age, what happens is the DNA methylation data are first used to generate proxies for blood biomarkers, and those DNA methylation proxies are then fitted to mortality risk to compose the ultimate algorithm. And these second generation clocks are substantially more predictive of mortality than the first generation. They also appear to be more sensitive to exposures that shorten healthy lifespan. Ultimately, we think they may be more sensitive to intervention. However, there are some limitations to methylation clocks. The first generation clocks in particular are sensitive to confounding by mortality selection, or sometimes called survival bias. So if you're building a model of aging by comparing younger people to older ones, you face the challenge that whereas people in their 20s and 30s are basically representative of the same population they were born into, but by the time you get out to your 70s and your 80s, roughly half your birth cohort has already died, many of them from aging-related diseases. And as a result, your attempt to estimate an aging gradient from the differences between these people is confounded by the fact that your oldest participants necessarily represent the slowest aging segment of your population. That can partially be addressed by modeling mortality risk instead of chronological age. But you then face this next challenge of cohort effects, which is that in a cross-section of data where you're looking at, let's say, mortality risk in people who are 20, 30, 40, 50, 60, the differences between individuals in age are entirely confounded by differences between individuals in the time when they were born. And when I say confounded, I don't mean that the associations are necessarily not aging. It's just that they cannot be distinguished from differences in the exposure histories of individuals that are determined by the timing of their birth. And these include things like exposure to air, for example, in which cars with leaded gasoline were driving around, pathogen burden changes, medication changes in the practices around childbirth, and so forth. And so we know that environmental toxicants and health behaviors have changed over time, and that these leave marks on our biology. And so we have the challenge of distinguishing between the biology of aging and the biology of changes over time in how people live. Finally, there's a challenge of when it is that this so-called age acceleration occurs. So typically, when people are studying epigenetic clocks, they focus on a measurement called age acceleration, which is the difference between the age we think your clock should read out, which is a prediction based on your chronological age, and the age it actually reads out. And those differences do forecast subsequent risk for disease, disability, and mortality. They're profoundly interesting in that way. But we don't know when those differences arise in the course of people's lives. And we still don't have studies, in part because we don't have that much longitudinal DNA methylation data out there, that establish how particular exposures may change, accelerate, or decelerate the ticking of these epigenetic clocks. So it was into this kind of area that we saw an opportunity to try and adapt our pace of aging measure into the methylome. This is what I just talked through. And so we developed a measurement that attempted to distill our 18 biomarker longitudinal pace of aging assessment into a single cross-section of DNA methylation data so that the pace of aging could be measured from a blood test. And we followed the methods pioneered by Horvath and his colleagues of using a machine learning analysis to fit DNA methylation data to a target, in this case, the pace of aging, the rate of change occurring across these 18 biomarkers over a 12-year period. And so our DNA methylation pace of aging measure is, in essence, a DNA methylation prediction of how fast the body has changed over the past decade, roughly. So in that way, it has the potential, at least, to be more focused on the exposure occurring around the time of measurement, and therefore potentially more sensitive in the context of evaluating an intervention. Just to sort of put a finer point on this distinction, epigenetic clocks ask the question, how much aging has occurred in this individual up to the time of a blood draw? The pace of aging, rather than a clock, is more like a speedometer. It asks, how fast is your body aging right now? And in our paper, we first established that the pace of aging could predict the same kinds of things that our physiology-based measure predicted. And here we're looking at DNA methylation data from participants collected when they were 38 years old, and we're looking forward to functional tests performed when they were 45 years old. And we see that people measured to be aging faster have poorer physical and cognitive function, and they show subjective signs of aging. That's the bar chart on the far left of the slide. And then the scatter plots in the middle of the slide are showing you association of age 38, DNA methylation pace of aging with change over time in the functional capacities that were measured in parallel at both assessments. So we have sort of proof of concept there that the methylation measurement performs similarly to the physiological measurement. We're also able to do some things with the pace of aging now that we couldn't with our original physiology-based measure, because we can take it out of the Dunedin study and into other cohorts. So one of the things we were interested in understanding was whether the aging rate increases as people grow chronologically older. This is predicted from demographic data on mortality. We know that the mortality rate increases with advancing chronological age, suggesting an acceleration of biological aging across the lifespan, but it's something that's not been testable with existing tools. And so these are data from the Understanding Society cohort. They show that there is a modest increase in the estimated rate of aging as we move from younger people to older ones. And of course, the caveats about analysis of aging in a cross-sectional sample apply. We may, kind of understand this as a lower bound of what the correlation should look like. This is an association with self-rated health, sort of replicating our observation in the Dunedin study in this Understanding Society cohort. So in this mixed age sample of adults, we again see that those with a faster pace of aging rate, their health as being poorer. There are also data in the paper showing that in the Understanding Society study, participants with a faster pace of aging had a physiologically predicted biological age that was older. This is an analysis of mortality. I'm showing you here the participants grouped into those with slow, average, and fast pace of aging, where we're defining slow and fast as just being a standard deviation above or below the cohort norm. And you can see that there's a non-trivial increase in mortality risk associated with an increment in the pace of aging, about 30% for standard deviation. And these are data from the normative aging study. So these are all older men at baseline, and nearly all of them have died over the course of follow-up. So it's an interesting opportunity to analyze mortality in a sample with complete information. These are some ongoing analysis that Miraj Kathari and my group is leading, looking at mortality in the Framingham cohort, a bit younger here. Only about 25% of the cohort have died, and fairly dramatic differences we see in the Framingham data. Those with faster aging are at nearly twice the risk of death over follow-up. This is more similar to what's been reported in other places for the grim age. Okay, so the measurements aside, and my time running short, let me get to what you actually came for, which is analysis of the CALERIE trial. And I apologize, I have the wrong header slide there for you. So this should have been bullet 5, analysis of the CALERIE trial. These are data that we published in our original report on the in the eLife paper, showing that in pilot data we generated for CALERIE, baseline methylation pace of aging was correlated with the future rate of change in that physiological measure of biological aging I showed you earlier. And that's what you're seeing on the left panel of the figure. Faster pace of aging predicted a faster rate of increase in that Clamera-Dubol biological age measured from the blood chemistry data. But that was not true in the caloric restriction condition. And so our interpretation of these data was that effectively calorie intervention had disrupted the pace of aging these individuals were experiencing. So in the AL group, we see that the predicted pace of aging holds over follow-up. But in the CR group, it does not. And so the ultimate question, of course, is what happens at the molecular level? Do we see those changes? And so with support from NIA, we've been able to generate a 3-time point DNA methylation database for Calorie. From the blood samples, there are data coming from the Horvath lab for adipose tissue and muscle samples on a subset of participants, and we're going to have gene expression information in those tissues as well. We had hoped initially to have gene expression data from the blood samples, but it turns out the RNA that had been collected by Calorie and stored had been corrupted over time, so we weren't able to generate those data. But we do have these DNA methylation data, and we can now ask whether calorie intervention changes the pace of aging measured at the molecular level. So just to orient you to what I'm going to show you, these are data that are being analyzed by my postdoc, Reem Waziri, and I'm going to show you 3 groups of measures. I'm going to show you the first generation epigenetic clocks. I'm going to show you the second generation epigenetic clocks. And then I'm going to show you our pace of aging measure along with a new measure that we hope to have out soon based on an updated pace of aging from the Dunedin study. And so for each of the clocks, the expected change that occurs is roughly 1 year increase in the clock age per 12-month follow-up in the Calorie trial. And I'm going to show you first the data from the ad libitum arm of the trial. So these are control participants, and then I'll overlay the data from the caloric restriction group in the trial so you can see what the treatment effect looks like. And so what we see is that there's clearly increase within individual in their clock ages across time. And for the Hannum clock, in fact, it's roughly 2 years over the 24 months follow-up of the trial. So the clocks are ticking. They're not ticking at quite the same rate for everyone. You can see the confidence intervals are sizable, and the rate of increase isn't necessarily uniform across the first 12 months versus the second 12 months, but it's happening. And that's also true when we look at the second generation epigenetic clocks. Morgan Levine's pheno-age clock on the left and Akelu's grim-age clock on the right. And again, note the variation indicated by the confidence intervals in how much people are changing. It varies between individual. It also varies a bit between follow-up interval. Now, the third group of plots I'm going to show you for the pace of aging measures, it's worth noting that the expected change in the pace of aging in the ad libitum arm of the trial is 0. So we don't expect the pace of aging to be modified for people who didn't experience intervention. The pace of aging is expected to be 1, 1, 1 at each follow-up interval. And that's roughly what we see for our original measure. For our newer measure, there is this increase that occurs in the AL group, although it's not statistically different from 0. So now I'm going to show you what happened in the caloric restriction participants. Am I? I'm not. I apologize. I'm not quite sure what happened to that slide. So what I have here for you are the treatment effects. So I will attempt to recover that information as soon as I finish the presentation, and I'll get that slide up for you. What I'm showing you here are the treatment effects. So this is the difference in the change occurring between AL and CR. So what we're expecting in all the cases is that the differences should be below 0. So the amount of change occurring in the CR condition of the trial should be less than the amount of change occurring in the ad libitum arm of the trial. So if the epigenetic clock ticks forward by 1 year in ad libitum, it should tick forward by less than 1 year in caloric restriction. And so what we can see in the case of the Horvath and Hannum first generation clocks, and also in the case of the Levine pheno-age clock, is that that doesn't happen. We don't see the expected difference between the caloric restriction rate of increase and the AL rate of increase. We do see some evidence of that happening for the grim-age clock, although it's not statistically different from 0. It is at least in the expected direction. And then for the pace of aging measures, At 12 months, we see the expected difference at both time points. At 12 months, we see the expected difference for both measures, and at 24 months, we see it for this new measure that is not yet out there in the public domain, but stay tuned. So I think we have here some evidence that we can recover at the molecular level a hint of the changes that were observed in people's physiologies, although clearly there's more work to be done understanding these measurement differences. I also want to foreground for you some complexity in the CALERIE trial. So these are the data I promised earlier on the adherence or the dose in CALERIE. So the box plots on the slide are showing you for participants in the ad libitum on the left and caloric restriction on the right arms of the trial, the distribution of percent caloric restriction relative to baseline that was achieved over follow-up up to the time of the measurement. So the 12-month measurements reflect the average CR through 12 months, the 24-month measurements reflect the average CR through 24 months. And what you can see is that on the right-hand side of the slide, obviously people's adherence declines over time. So it was hard to get to 25% CR, and it was hard to stay even at a lower level of CR, but also that participants in the ad libitum arm of the trial undertook some caloric restriction in some cases. And so there's this kind of contamination of the treatment that occurs. And so we applied another analytic strategy to try and get past this, using an instrumental variables approach to decontaminate the treatment effects. And this estimates what's called an effective treatment on the treated. Essentially what we do is we build a first stage model that predicts the change in caloric restriction that was caused by the intervention. And we then replace the sort of intent to treat dummy variable of intervention condition with the estimated additional percent CR you undertook because you were in the intervention group, net of your pre-treatment characteristics. And what we can see from this analysis is that it doesn't really change our results for the first generation clocks or the pheno-age clock, although again, it's clear that, although the change is not in the direction we expect, it's also not different from zero. The effect for the Grim-age clock is slightly larger. I apologize, these graphs are on a different scale than the ones I showed you before. And so in fact, the Grim-age clock effect size is bigger in the instrumental variables version of the analysis. And that's also true for the pace of aging measures. So this instrumental variables approach allows us to estimate, and sorry, here I'm showing you effect sizes estimated as if everyone had achieved 20% caloric restriction. So not quite the prescribed dose, but somewhere close to it, something that we observe not infrequently in the intervention group participants, especially through the first 12 months. This is what the effects would have looked like if everybody had adhered at 20% in the intervention group and they had maintained their same caloric intake in the control group. So with that, I will leave you with a couple of observations, namely that the projection of these measurements of biological aging into randomized and controlled trials is the next frontier, in understanding the feasibility of intervention to extend health span. Clearly, we have some distance to cover in our development of measurements. I know that Morgan Levine's lab has some refinements to the existing clocks it's about to publish. Steve's lab has new clocks, we have new measurements, and this will no doubt continue. All of this should be considered with the caveat that we're still training and validating our measures of biological aging in samples that are overwhelmingly white, and in some cases more educated than we'd like them to be. That's not true of the Denise sample, but it may be true of some of the other samples that are being used. And so diversifying the data sets we're using to develop measurements of aging will be key in ensuring that their utility to clinical trials sort of maintains our priority for interventions that build health equity rather than those that work only for the folks who usually show up in our trials. And then finally, I think partnerships between people working to develop measurements, people studying longitudinal cohorts that are able to observe the aging process, and interventionists are all required to get us where we need to go. So there's a PDF of these slides that will be available. I'll make sure that the right information is there. And then the code to implement our pace of aging measure is available on GitHub, and you'll be able to get that link out of the slides if you need it. So thank you again, and I apologize for running a bit over my projected time, and I'm gonna work on, while I stop to share, pulling up that slide that I had promised you about the change in biological aging in the caloric restriction group. So thank you very much.
Thank you, Dr. Belsky, for that excellent talk. Your amazing understanding from the cellular to the epi level really made this a fascinating discussion, and all the data that you shared is just so interesting. We'll start with our Q&A. And the first question is from our center director, Dr. Sarah Booth. She asks, with the COVID-19 pandemic, how will these algorithms address the complexity of stress, illness, and other exposures of the virus on aging predictions?
So the question again is, how can these measurements of biological aging inform our understanding of the impact of the pandemic and maybe our efforts to intervene? This is a really interesting question. So I think that one of the things we'll be able to do in longitudinal studies that have collected measurements pre-pandemic and then post-pandemic is to quantify in real sense how the pandemic has affected the rate of aging in the population. We know that it's shortened life expectancy because it's killed a lot of people. But I think we'll be able to forecast with these kinds of measurements what we may expect to see in the future. And there are different pathways through which the pandemic may affect population aging. It may be the case that people who have been infected and have recovered nevertheless sustain insults to their biology that lead them to have a more advanced biological age, or even in the future to be aging faster. But it's also the case that the period of lockdown has imposed dramatic stressors of a variety of kinds on people, and we may expect that those pressures have also accelerated the pace of aging. And I imagine that with the data that have been collected, we'll be learning more about this soon. I should say that Morgan's lab has a paper out in preprint. I think it should be published soon, with the interesting observation that a blood chemistry measure of biological aging predicts in the UK Biobank who was more likely to develop severe COVID-19 infection. And so that sort of validates a lot of the editorials that were written early in the pandemic about how this is clearly a virus that is disproportionately affecting older people, but not just based on their chronological age, based on their biological aging. And so interventions to promote healthier aging and slow biological aging may also build resilience against not just this virus, but viruses to come.
Great, thank you. The next question is from Dr. Alan Taylor. He asks if you could correlate Gero genes with the rate of aging as per your clocks. So can you correlate?
Yeah, so the question here is, we have, there are lots of observations, especially from experimental models, that genetic differences affect the rate of aging. And there are some data in humans that suggest the same. We now estimate that perhaps only about 10% of variation in human longevity is attributable to genetic differences between people. That's down from 2 or 3 times that number, a decade ago. But genetics definitely play an important role, and I think it's absolutely the case that as an endpoint in, for example, a genome-wide association study, there's a lot of promise for these measures of biological aging. And that's something that we may see as we have, in particular, DNA methylation data at larger scale. So to do these gene hunting studies, you need at least tens of thousands of individuals, possibly hundreds of thousands of them. And so we're still a long way off from that and having harmonized databases, either of blood chemistry measures that would allow us to estimate biological age, or DNA methylation, or whatever will come next. But I think it's definitely something interesting to investigate, and it may be possible that focus comparisons of already identified longevity genes, I think as the questioner was suggesting, have some promise there. And so these sort of emerging multi-omics data sets that allow us to put together DNA methylation along with DNA sequence may provide a path forward for that.
Great. The next question is from Dr. Shandong Wang. He asks, in your pace of aging study among the 26 to 38-year-old, have you also examined some of the early life risk factors such as smoking, including passive smoke exposure, alcohol drinking, and could that predict the accelerated pace of aging?
Yeah, so we've not looked too much at health behaviors specifically within the Dunedin study, in part because those behaviors would be observed around the same time we were measuring aging itself, and so we wouldn't have good separation of exposure and outcome. But I think that's absolutely, a hypothesis we would hold. I know there are some papers in under review now looking at alcohol exposure, and we actually do know something about obesity and tobacco exposure. Both of those are associated with older grim age, faster pace of aging, older pheno-age clock values. And we've also done work on social adversities. So, I showed you the data, the physiological data on how growing up in a poor family might be associated with a faster pace of aging. But we've also reproduced those observations with DNA methylation measurements. So kids who grow up in poverty tend to, as adults, be aging faster than peers who grew up in wealthier households. It's also true of kids who were victimized. And now there are data coming out linking kind of adult socioeconomic circumstances with a faster pace of aging. The Life Path Consortium has published some data on a number of these associations, so smoking, tobacco, dietary behaviors, and the epigenetic clocks. There is a paper, the first author is Fiorito, 2019, in the journal Aging, I believe, that summarizes data from the several cohorts that they've harmonized for that.
Yeah, we're at 1 o'clock, but I do want to continue with a few questions, and we'll have the recording for those of you who have to leave. So the next question is from Dr. Christopher Wiley. He asks, you've acknowledged several of the key issues surrounding methylation clocks. There also appears to be a confirmation bias surrounding the techniques, example, sample types, and interventions that move the clock in unexpected ways, and those do not get published or are published in lower impact journals. Of course, this is not unique to clocks, but without mechanistic rationale for changed methylation site, what would we say that the clocks actually predict?
So this is an excellent point. I think it's one that the field has gotten much better at dealing with over the last few years. So early on, the file drawer was just full of papers finding null associations between the epigenetic clocks and you name it. As data have become, methylation data have sort of proliferated, more observations are getting published. And as the clocks have improved, people have been braver about reporting associations for multiple clocks. And I hope that we've had some part in that. It was very hard to get our paper reporting these very weak correlations between the clocks and the physiology measures published. It took us a year and a half. But since then, I think a number of papers have come out to show this. So I think we're getting better at opening up the file drawer and being honest about what is associated and what isn't. And I think that, sort of the general process of educating researchers about, what are best practices and journals about, what their responsibility is to publish is having some impact. I would say that there has always been, from the beginning of sort of the clock making business, a concern that the absence of mechanistic understanding of what biology the clocks are actually representing, sort of renders them uninterpretable. And I don't think they're uninterpretable, but they are a mystery. We do want to know what makes these clocks tick. Ultimately, I think the way to figure that out will not necessarily be precise understanding of the mechanisms of the individual CPG sites that are represented in the clocks, because machine learning just grabs variance that is reliably measured and relatively independent of other reliably measured signals. So there's not necessarily real biological substance in any of the individual CPG sites picked up by the clocks. They're just correlated with something that's biologically substantive. The path I think we need to go down to understand what makes the clocks tick, or what makes the pace of aging speedometer go high versus low, are experiments. Experiments with human participants like caloric restriction, drug trials like the Fahey trial that was published, although ideally with a control group, but but also studies that can happen in dishes. So Martin Picard at Columbia has this study of cultured fibroblasts in which they're observing epigenetic aging within cells and then administering various interventions to those cell cultures in ways that allow them to understand how different kinds of microenvironments may affect the ticking of epigenetic clocks. And so I'm more inclined to kind of work in that direction rather than in attempting to identify CPG sites that we think have biological meaning, not least because we're using CPG sites assayed principally in leukocytes to infer the state of biology throughout an organism. And that sort of puts you pretty far afield from what I think most bench scientists would recognize as actual mechanism.
Great, thank you. And we'll take this one or two more questions and then stop. This is from Dr. Alice Lichtenstein. She says, thank you for your fascinating presentation. With regards to the calorie study and improvement in measures of cardiometabolic risk and aging, could the difference in diet quality potential improvement that is be separated from overall change due to calorie restriction?
So this is a really good question and an important one for the trial, Sai. As you well know, there is extraordinarily detailed information about what the participants ate. But those data have not yet been sort of thoroughly sifted in ways that provide us with good phenotyping of the construct of dietary quality. It is the case that in Calorie, the goal was kind of macronutrient intake reduction, right? So it was about getting calorie counts down. And while there were a series of different diets offered to participants to achieve that when they enrolled, those who found that initial enrollment diet, sort of unsustainable, were allowed to switch. On the one hand, that's a powerful opportunity for within-person analysis of how changes in dietary composition affect their physiology, but it also makes it really hard for us at a sample level to try and disentangle the specific components of diet from the sort of time period where people were in the trial and their total caloric restriction. So excellent question.
Yeah, and along the lines, Dr. Mazepin asks, do you think that carbohydrate restriction could, elicit the same effects? And I guess that is pretty much also, something that remains to be answered, right?
Yeah, I think that's right. There were low carb diets within Calorie, and so it's possible that at some point we'll be able to, sort of compare total caloric restriction to carb restriction and estimate effects. I would say that, if we're thinking about the measurements I showed you today as our kind of surrogate endpoints for health span extension, we're not going to have the statistical power we need to make those fine-grained distinctions. But the great thing about these omics databases that we're generating is that they're not just good for the measures we have today, they're good for the measures we're going to develop next year and the year after and the year after. So this data resource will only grow in its richness and its power as we learn more about how to measure the aging process. And that may ultimately allow us to revisit some of these questions with greater precision.
Great, thank you so much, Dr. Belsky. This was a fascinating discussion. We have a few more questions. We'll have you stay, if you don't mind, and we'll have those answers posted for our scientists and other members of our community to be able to have those be answered. So thank you again, and thanks everyone for your attention and for staying over.
Thank you.